All right. I guess we'll start with a little bit of the housekeeping rules, and we have 31 people so far, so they can all join in. I would just want to say, everybody can hear me, hopefully. Thank you for joining us for a review of the 11th International SDS Scientific Congress. We are so happy that everybody could be here and you could join us. Just a few housekeeping rules. We're going to mute all. So if you guys could just please keep your microphones off while Dr. Rommens is giving her presentation. If you have questions on the bottom of the screen, you'll see a chat box. If you click on it, it will open a box and you can actually type your questions in there. This way we can keep them. And if we don't have an answer or we need to get an answer, we'll know who asked it and we can always get back to you later. And if we don't have enough time, then we can always, type them up and answer them later. We are recording this. So once we're done recording it and we get it out there, it'll be out there on YouTube. We'll send everybody the link so that if they want to watch it again, it'll be out there and we can always go back to it. So I guess I'm going to introduce to you guys, if you don't already know her, Dr. Johanna Rommens from the Hospital for Sick Kids in Toronto, Canada. She's also the chair for the medical board for SDSF. And just a little bit I found about Dr. Rommens is a world-renowned geneticist and molecular biologist whose groundbreaking research has had a profound impact on the field of rare genetic diseases. She is best known for her discovery of the SDS gene, the gene responsible for Shwachman-Diamond Syndrome, and a pivotal finding that has shaped the scientific understanding and diagnosis of SDS worldwide. In addition to her work on SDS, Dr. Rommens has contributed to discoveries in cystic fibrosis and other inherited conditions. She continues to lead cutting-edge research at Sick Kids, although I think she's partly retired, I'm focused on understanding the genetic and molecular basis of disease. So if everybody would please welcome Dr. Rommens, and she's going to share her screen and show us a bunch of complicated stuff.
Okay, so thank you everybody for joining us today. I'll do my best to describe to you all the exciting things that happened at the Congress, and in a few places I'm going to squeeze in a few little bit of background things that I think will help give context. At least that's my plan. So now I'm going to share my screen so that I hope you can see my slides. Can I get a yes on that from someone?
Yes, we see
Okay.
research update. Yay.
Okay. Okay, fantastic. Okay, can you see my laser pointer as well?
Mm-hmm.
Yes, I'm getting a nod. Okay, so I'm going to try to describe some of the things that happened at the Congress and also recognize any kind of confidentiality there may have been for some of the participants. So the way,
Oops.
oops. Okay,
Sorry. There we go. I hit the buttons. Why is it?
so what I'd like to do is I'll have to I grouped a lot of things to give you to try to give and try to keep that there was still some logic. So first, so the symposium was held in June, in early June. We had 3 full morning and 3 full afternoons, each with 2 or 3 events, and that included things like lecture sessions where there were speakers. Some sessions were more discussion based with guiding moderators and ability for audience to interact. Had also a very special lecture by Dr. Shimamura from Boston. So this was sort of the keynote speaker of the meeting where she described her work and her interest in Shwachman syndrome and so on. And also we had a poster session, I think it was on the Friday, where we could all meet in one-on-one with the the authors of the submitted abstracts. We had one special social, well, there were a few special social events, but a key one was the dinner on Friday night at the Cincinnati Museum Center, which I heard was an old train station. The neat things there that I noticed were the dinosaur skeletons. And there was also a rock from the moon landing from 55 years ago because Neil Armstrong home is in Ohio. And in fact, the rock was kind of small, but it was still special because it was 55 years ago, just a week ago today, that we landed on the moon for the first time. And I actually remember that day. So it was special to me. So that was great. So let's get on with thinking about or talking a little bit about what things happened at the meeting. So one of the events was these opportunities that I said for interaction with the audience. And we heard some comments from various family members about things they'd like to see and things they deal with living with SDS broadly. And there were a variety of things, and I'm sure you can appreciate some of these. And I'll try then from these lists, I'll try to show you that I hope we've been able to help some of these kinds of things. There was calls for having more adult care and having physicians trained in adult care, which we appreciate is sort of an ongoing challenge for many families. And this point, though, also emphasized that we have to think of care that extends over a lifetime of any particular individual with SDS. There was also some comments that it was really helpful to be sure to give patients some education regarding their SDS, their conditions at age appropriate, in age appropriate ways, and also at age appropriate times that would help them, give them some ability to participate in their care and in appreciating and making decisions day to day. There was some concerns expressed for excess and medication costs. Certainly, probably one of the bigger concerns one here is the challenge of uncertainty every day, and it was certainly helpful to always remember to try to help with coping. It was also that all of these things together to appreciate. There was a request from some family members to think about the variability between patients. This is going to be a theme that will come up. It is a challenge in Shwachman syndrome. It is a challenge generally in a lot of genetic diseases, but it's especially, I think, a challenge in Shwachman. And also recognition of everybody's different and difficult journey in their lives. And that was things that we wanted to think about. So that was one of the discussions that we had. We had another discussion asking the audience where they thought research should be headed in terms of where it's going. Well, one of the things that came up then was, please, we need consistency. That is, patients and families needed to receive consistency, and that's certainly a value that I can appreciate that is very important. There was calls to get more investigations for patients who have unknown genes to date that do have SDS clinically, but it's not clear what is the cause. There was requests for investigations into features on non-hematological features. Well, hematological features are a major point, but also to think about the other organs that are affected. This is a syndrome, so one needs to think about the other organs that are affected. Pancreas, the liver, and especially what happens with these organs through life, through one's life and into adulthood. More on being able to cope everyday issues, the practical things like being able to eat and how to use the pancreatic enzymes, thinking about when are vitamin supplements necessary, also living day to day with some neurobehavioral features, and also concern about some social interaction or some challenges with social interaction, part of it due to chronic illness, part of it due to having Shwachman. Also to appreciate and understand anticipation. Is there ways more could be done to anticipate when to worry about the bone marrow failure and for the cancer risk? And certainly this was a big question that got a lot of attention at the meeting. I'll come to some of these points coming up. And also, would there be a way that not only could we anticipate them, could there be development of strategy to think about that? And so that was appreciated what the plan was. And idea was where one could be somewhat proactive and not just react. And that kind of made sense. And this is especially prominent when thinking about the hematological features of the disease. So one of the things that got a fair bit of discussion from a number of speakers was the issue of care over major life transitions, and particularly in the real world, this happens pediatric to adult, but this also happens in other ways, in social ways, when a child grows up and goes off to college or goes on and goes out to work and does other things. So these are all changes that happen and that might require changes in the care team, for example. Certainly that happens between pediatric and adult stages. And it's certainly aware that different centers might manage this differently. But certainly there was encouragement to always try to build relationships and also to kind of enforce, at least encourage that any kind of transition like this is not something you want to do in a moment. You want it to be proactive, you want to have a phased approach, and you would like that there is some overlap in time. So, for example, one physician commented that the best thing was to have, for a time period at least, that perhaps the patient sees both the pediatric and adult care settings over a period of time, and then there's sort of this becoming familiar and then also sort of a letting go, and that made a logical evolution for that particular person. The other thing that was encouraged, and this was encouraged from physicians who do provide care, and that was to encourage new care providers to learn. And certainly it was pointed out that for some patients, it was felt that if the pediatric care had been strong, that that was the best segue into getting good adult care as well, if that was possible. And we learned of a few examples and of how this is being managed. It's very different in different places, as I've alluded to. In Philadelphia, for example, they have a combined pediatric and adult Center for Bone Marrow Failure Syndromes, which is probably quite the extreme and not typical in most hospitals for all kinds of reasons that might not work everywhere, but this seemed kind of a nice resolution to issues. There were comments from physicians from Germany, for example, that some of their special care team specialists actually transitioned themselves from patient to an adult clinic where the doctor just went to the other clinic. And that they were certainly encouraging that kind of approach because that made it easier for people undergoing life changes and things like that. So that was actually quite interesting to think about, and certainly an area where there's issues to deal with that. It's not easy for everybody, but things that we'd hope could be managed a bit better for many patients. Okay, so these were the discussions with the audience, and I want to talk a little bit more now and go a little bit more to sort of the more basic aspects. So I'll give a little bit of introduction. So I probably don't need to spend too much time on this, looking at our favorite gene, SBDS, which causes about 90% of SDS cases. So this involves loss of function variants in this gene, and from the literature, there's about 100 distinct causing variants, although there are some that are more common than others. There are additional genes also implicated, and I hope I'd be able to convince you a little bit why it's kind of logical that they might cause a similar disease. I hope I can impress upon you, certainly for EFL1 and also DNA JC21. And also we have very like syndrome with variants in the SRP54 gene that has been seen. So on thinking of these genes, all of them play a role in either making proteins or building a ribosome, and that's why we think of SDS as a ribosomopathy. But I want to just go back a little bit and remind you about the ribosome, because it's from the ribosome we need to learn what is at the molecular level the failure. So this is a picture showing the maturation of the ribosome, and just to give you some context, here is the nucleolus inside the nucleus. And so there's a cell here, and this is in the cytoplasm. And to remind you that the ribosome is one of the biggest working machines in a cell. It consumes amongst the most energy of any part of the cell. So a cell has to commit a fair amount of effort to this. There are at least 4 ribosomal RNAs. There's 76 small nucleolar RNAs, 80 ribosomal components, but it takes over 200 factors to assemble the ribosome. Okay, so that's a lot. So when we want to think what's happening in SDS, we want actually to look, once that pre-ribosome segments are made, they go into the cytoplasm, but there's still maturation steps that happen. I'm particularly pointing to the large subunit here because that's where we see failure in SDS. And I want to look at that now a little bit more closely. It's shown here one step from this 6DS premature ribosome to the mature ribosome, or mature large ribosomal component. But if we look a little bit closer, there's actually multiple steps, and this is looking at the steps. I purposely showed this complicated picture to impress a few things upon you. First of all, building a ribosome is quite an amazing event, and we're looking here at real pictures that have been captured, both from structural information of proteins, and the proteins are color coded. Um, and you can actually see them. And so we're building the 6DS ribosome, and all kinds of things have to happen. New proteins have to come on. Some of the assembly factors help. Some of these assembly factors come onto the ribosome, but then they leave. But they have specific jobs to do. So, for example, they could maybe give energy. They could supply energy. They could provide an adapter that lets a next protein come on. And what I want to impress upon you is the multiple steps, and also that some of these are consecutive steps. Each one of these pictures is looking at the same face of the ribosome. There are some things we can see about ribosome parts that we know. The stalk, the L1 stalk is here. At the bottom here is what will eventually become the peptide exit tunnel. That's a specific part of a ribosome. So we're looking here at all the parts, and you can see like the ribosome, 6DS ribosome gets bigger because of the addition of this particular gene. And then it gets taken away, and that allows other things to get added, and you're seeing new things added. For example, from here to here, we're seeing this new gene get added here, this LSG gene. And I should point out here, I'm looking at names of the yeast ribosome. It's ribosome yeast are quite different than us, but the ribosomes are actually not so different. So we can draw our attention to them. And I want to remind you who these players are. We know a few players that we care about in SDS. There's SBDS, we'll come to that in a moment, but there's also players TIF6 from yeast and EIF6. That's this pink one here. And you can see that he's always hanging on to this maturing ribosome here. Then you see this new purple one come on, for example, and he stays on. So these are that latter steps of the maturation that are occurring, but we need to look a little more closely at this to see additional proteins play a role. Oh, and one aspect of these exquisite images that are showing changes in the ribosome, I want to kind of give you a way to think about how these changes are occurring. I want you to think about a key and a lock. Because what has to happen for a part to get added, it's like you have a key, it has to fit into a lock. And when that happens, subtle things can happen. The neighboring protein, for example, can shift slightly, and that might open a new lock to which a new key can enter. So some things have to happen consecutively, but it's a busy place. This is a big machine. There are 80 constituents to the ribosome. There's stuff happening on the front here. There can also be stuff happening on the back. So some things are simultaneous, but some things are consecutive, particularly the idea of that key and the lock. And then once that key fits in, you might open a new lock. A part might have to follow away to allow that lock to be visible so a new key can come in, where the key now is like a protein that's coming in, some of the proteins that's building, and that's how you build a ribosome. So these are the steps of the ribosome as it comes out of the nucleus into the cytoplasm. I want to pay particular attention to these latter steps here because I'm going to look really close at the players here, but I'm going to change the graphic here, and I'm going to take a graphic from one of Dr. Alan Warren's review papers where he's looking at that. But you'll see some players you should recognize. Here is SBDS. Here is the protein EFL1. SBDS and EFL1 work together. Here is, and what should happen with these two, and now we have keys and locks here, SBDS and EFL1 comes in, and when that happens, we can kick off this player EIF6, okay? And that has to happen to give us the mature ribosome. And there's a few more players here to call your attention to. There's DNA JC21. That is also a causal gene that leads to SDS, and you can see it's just a few steps ahead. So it's similar, and it is involved in maturing this protein, or the ribosome machine. So it makes sense, I think, to think that if we interfere at a later step, if you just interfere just a step or two ahead, and remember this is a process with like 200 steps, that you're going to see something potentially similar. So that's something to think about in terms of what's happening and how these proteins might be working. There is one protein that's not here, and this is the protein that causes an SDS-like syndrome, which is SRP54. And I, just for time, won't have too much time to talk about it, other than to say that it does take part in the ribosomes, but it's at a different step in terms of where its role is thought to be. So, but what happens? Let's look a little bit more closely. I'm sure this is my drawing, so it's not near as elegant as the other pictures. But we're looking here at the 60S large part, the 40S small component, and these have to come together for you to be able to read a gene, and from that you can build a protein. So remember, your cell has about 8 or 9,000 genes that it reads, and into proteins, they'll have 8 or 9,000. Some of them are the ribosome particles itself, but some of them are just ordinary things that you need. So, for example, if you're in the pancreas, well, you need digestive enzymes, and only the pancreas, the exocrine pancreas, builds your digestive enzymes. So this messages would code, the messages that get read from the genetic code in the pancreas are the digestive enzymes. And there's about 28 peptides. Not all of them are digestive enzymes because, as it turns out, digestive enzymes are a little bit toxic things. And you have the protein of the digestive enzyme, but you also have to have chaperoning proteins that hide the digestive enzymes because they're quite toxic. So you don't want them made anywhere else except for in the pancreas, and you don't even want them working in the pancreas. You want them to get to the intestine before they do their jobs. But so what has to happen in typical translation, we need to kick off this EIF6, and that's done, as I pointed out previously, by SBDS and EFL1 working together. And when that kicks off, we know that this can join and you get the ADS ribosome, and that can now be a working ribosome. And that's typical translation. However, if SBDS is failing in some way, that can't happen properly, and therefore you don't get an intact proper ADS ribosome, and you don't get translation. In fact, it's true if we look at all of the genes that have been implicated in SDS, all of them with their loss lead to much less protein translation. So you're going to make a lot less proteins in a cell if you don't have these particular factors, and that's partly because the last steps of degeneration of the 60S component is failing, and that's a problem. So as it turns out, it's a problem for overall translation. What we're not really good at yet is knowing if it affects every gene that gets translated or whether there's subsets of them. I actually think that it's probably a little bit different in some cell types than others. And so we need to understand that better. We don't understand that perfectly well. There are some ideas out there, but we also have to think that once translation is done, once that message is done, the ribosome actually falls apart, the two subunits stay off, and actually to a large extent will recycle. So, but if EIF6 doesn't get knocked off correctly, what happens? These just hang around. Well, there's a little bit of evidence in some cell types they do just hang around, but in other cell types it maybe they get degraded. It's not absolutely clear. Now, if you think about this deficiency, we've actually learned some very interesting things about how, in fact, and this contributed to knowing how this got put together and how even the last steps worked. One of the things that it was work from Dr. Warren's lab who pointed out that if you take the SDS deficient yeast, so this is a yeast that's missing this gene, which is the equivalent to SBDS, it turns out that there is one way you can get some translation. And in fact, the yeast grow faster and have a reasonable amount of protein translation if you mutate this gene. So now this gene, SBDS and EFL1, have to kick this off. But the other way is if you weakened the ability of this particular factor to bind, okay? The key's not going to work as good in the lock as it was. There's benefit, and we know that because we see the yeast cells grow better. The other thing this is reminiscent of is there's additional evidence that this EIF6 is important in the bone marrow and blood of patients because, in fact, some patients in their bone marrow and in their blood have changes in their DNA that actually decrease the copy of EIF16. So how can we use this in some way to try to learn to think, okay, is there a way to alleviate some of the problems? Well, nature's kind of telling us something here that we should pay attention to. First, we know that we could interfere perhaps with the interaction here of what's going on. So our keys and our locks, we want to think about them. How do we make them work and how do we know? But to be able to do that, we need exquisite information on structure, which is why I showed you those very detailed structures, because we do need to know precisely how to think about that. And in fact, Dr. Warren is kind of thinking about that and working. He's trying to think of a new key to change what happens here? And can you make a key, for example, that either weakens EIF16? Can we change the function of EFL1? Can we a little bit to try to change what's happening here when SBDS is missing, for example? So that's a kind of a very interesting approach, but it's a potential way to think about developing therapeutics. It's not easy, but just because it's hard is not a good reason to try. You might want to try to figure it out, but it's very challenging. And there would be a number of things to think about here with respect to what we've learned to how can we bypass if we don't have SBDS, for example. We can think similarly for the cases that are due to EFL1 and also for DNA JC21. That's one step back. We need to understand that one. Less work has been done on that to try to figure out how that might fail. But these are all things we could think about. So I'm thinking, though, about ribosomes, and I'm still thinking here at a very basic sort of level. You know, the interesting thing is that ribosomes are the most common working machine. It's present in all cells, like even cells undergoing division and all kinds of drama, they still have ribosomes and they're still mostly working. What we do know is that tissues express different subsets of genes, though. So, like I said, the pancreas has to make digestive enzymes. No other cells make digestive enzymes. The liver has to make its favorite genes. Blood cells have to make their blood cell growth factors and components that do all kinds of things. They make antibodies, all kinds of things they have to be able to do. They do, though, have different subsets of genes, and they also have different growth rates. And so this has been a thought for why perhaps different cells and different tissues may have different levels of ribosomes, although it's a mystery that you can find work 50 years ago when people were thinking about it. Why do some cells have more ribosomes than others? And what we've learned, and at one level, you'd sort of think, oh, well, it must all be coordinated and it must all be working very nicely together. But it's been a bit surprising to learn that the ribosomes are not that static, because you would think that if this is a machine that needs so much energy from a cell that would at least try to coordinate things. And it's true that in some model organisms, we see better coordination. In human, it's not so much, partly because we're multicellular and partly because our tissues make very different things, probably. So things are not fully coordinated. But where you especially see some drama is in some kinds of cancers. And I'm thinking now cancer not limited to SDS. Okay. So for example, for a gene like ZNF622, and I didn't point it out, it was on the previous slide. It's also at the very late stage. It's just before SBDS and EFL1 might be working, or even EIF6, EIF516. We think of that as being close to SDS and in leukemia, but it's also been seen that these two genes are actually quite abundantly expressed at very increased levels in other types of cancer. And what's interesting, you can think, well, that's just spurious because things go a little wacky in cancer, but that's not quite true because they're seen in each of these is seen in their favorite common cancers, and they're seen at the common stage of the development of that cancer. So that's more supportive of they're doing something different there or doing something important there, and that's what we want to think about. So interesting things about ribosomes. We want to keep all these things in mind. Other weird things about ribosomes that we know. Why is it that in the ribosomopathy diseases, we have failures in different components? Sometimes you see really only stark issues in limited tissues, and sometimes the tissues are shared across the diseases, but sometimes they also involve different groups of tissues. It's generally true in a lot of the ribosomopathies, and even in genes that have been found to be part of the ribosomes that have been found in model organisms, even if they've not been seen in a human disease, for example, lots of times neurodevelopment is affected and so on, and that's because the brain has particular need for high protein synthesis at certain times, at least that's assumed why. But not always straightforward, and it's not always logical. We don't always quite know what's going on. We also need to think about whether incomplete ribosomes or even individual factors have other functions. And the most glaring occurrence of this that we know of in nature is what viruses do when they infect cells. They can, in fact, hijack what some ribosome proteins do. In fact, I was reading, and it's not true for the SDS genes, but for some of the other ribosome cofactors, it turns out COVID binds them and uses it for their replication, which is kind of weird. But that kind of suggests the idea that maybe some ribosome proteins have other functions, and that's a bit weird. We're not quite got that all figured out yet. But when we think of ribosomes, we want to think about how they're regulated, at least sort of from three angles. First, how many ribosomes in a cell are being made? Well, it probably has something to do with how much they need, but we don't generally always know that. How many are working at a specific time? And I can tell you from very old times, from old genetic data, looking at um, ribosomal proteins in flies, for example, we actually could see that some organs have all their ribosomes working all the time, full tilt, and other organs looks like half their ribosomes aren't working that tough. But stress them out a little bit and maybe they get engaged. So there's this variation that we need to think about. The other thing to think about is how many are turning over and getting degraded at a particular time. Like what do ribosomal proteins do? They're probably pretty stable as long as they're in a ribosome. But when the ribosome falls off the message and it doesn't get recycled or something, we presume it gets degraded because that's usually what happens with proteins. But we don't really know that well. The other thing, of course, that we want to think about is that since protein synthesis is such a major player in the cell, how is it linked to various cell stress responses? We know that proteins have to sense internal and external stimuli. They know and they want to grab the good nutrients that are coming, but they also have to get rid of waste, metabolic waste things that aren't good. If you drink a lot of wine for supper, like your poor liver has to take it because it has to get rid of some of that stuff, so it's got to get rid of it. But the cell and the proteins in the cell also confront other kinds of adverse exposures that aren't always so apparent. If you were in the sun this summer and got a little bit of sunburn, what happened is, well, your keratinocytes in your skin weren't happy about that, and they would have changed their protein expression and made things change, and ultimately you get your sunburn. And then if it was a really too long, maybe you've even your skin peeled, for example, and those are cells that actually chose to die, and that was their response. So that's a pretty dramatic response, but that can happen to cells. But we want to think about how this all works together, and I just gave you a long list of weird stuff about ribosomes that we really haven't been able to answer yet. But there is coming a way to answer it, how to begin to think about all these observations that I've referred to. And in fact, there's a whole emerging field on this idea, although this study has been around, and that's just thinking about how proteins are homeostatic. That is, how do we balance the amount of proteins in our cell, which is intimately related to how many ribosomes you have. And that field is called proteostasis. And there's all kinds of parts of proteostasis that we've learned. We've especially learned in recent times. So these are all the pathways and the networks that work that let you respond to external stimuli that may represent old and tired proteins that don't want to work anymore, or maybe you need to now recycle them, all the things that you have to do with proteins that are made. And we've actually learned a lot about this process of proteostasis, which holds the balance, in fact, for what proteins are doing in the cell. And we've learned a lot from aging, and we've also learned some of this from cancer. And they've been really, this idea and this concept of thinking about what's happening has been quite insightful to some of the process that we realize with what can happen with proteostasis and what controls the balance of proteins within a cell. And of emerging efforts have been the idea to start thinking about the proteostasis processes in stem cells in particular, because it turns out stem cells are kind of unique in some ways. And we suspect that their proteostasis process is in fact quite different than terminally differentiated cells or cells that are developing. So there was some talks on that, and I've given a few references here just to give you a little bit more of an idea, because that was certainly an emerging area that we heard a little bit about at the meeting. So that was quite interesting. So these are the kinds of basic things that we've been thinking about and learning about. So we heard a little bit at the meeting about model organisms and how they might be useful. So there's sort of been the traditional models in SBDS, EFL1, these genes now, the causal genes all have some animal models of some sorts that have been studying. And the first thing we've learned about them is what do their gene functions? What are they? What happens when they're not there and are not working correctly? And several of these are already published, so I won't dwell on these. And I just want you to, there was one really, there's some new things happening in the world of developing models with models on the horizon that I want to refer to. When you think about it, well, one way we study one way when we study models or diseases all the time is we just grow tissue culture cells in a petri dish and we look at them. Well, that's just cells growing on plastic. So that's kind of a static world. We also give them every nutrient we can think of, they'll make them be happy. But in the real world, it's not quite like that. You know, cells are competing for nutrients. They need blood to supply them with nutrients. They might have a cell that they like to be next to and kind of thinking like bone marrow cells in the bone marrow niche and that kind of thing. And that's the real world, not this growing it in a plastic kind of dish. So people have come up with, there's this new idea coming up with so-called organs on a chip, and that's actually what's illustrated here. And people now are talking about making organs on a chip. And here's the chip, okay? And so the idea of this chip is right here in this central part is where you'd put your cells. But you can build in various ways so that more like a tissue, as opposed to just growing cells on plastic, you might have multiple layers. You might have access, and that's what all these little blue heads and red heads are, where there's actually, you can actually see there's a fluid path. So for example, you can do all kinds of things for these cells. You can give them different nutrients at different times. You can take things away. You can wash them at different times. You can do all kinds of things. And that's a really oversimplified version of how this works. But this is a very fascinating new field of engineers of engineering to try to think about how to build um, organs on a chip. And this is a very fast-moving field now with lots of potential and in development. And in fact, the idea of trying to be able to go blood and or bone marrow cells and blood progenitor cells and to recapitulate the bone marrow and the blood cell lineages has been actively pursued by many groups that try to think about how to do that. Particularly looking forward on this would be because this is actually considered sort of micro scale, so it's very small. You could think about, is this a good way to do screens for drugs or potential drugs? And that's one of the things because it's a little bit more real, if you will, than just growing cells in plastic. So there's been this idea that this might be a more accurate way. It's possibly more convenient than models because as lovely as models are, they're never perfect. And it's true that a mouse does have SBDS just like human, but not everything about a mouse is the same. Like they make proteins for whiskers, humans don't. But there's other differences too, of course, that we'd want to think about. So this was kind of an emerging idea, which I thought was quite interesting and an aspect in development that I think as we look forward to in future years, we'll see some things happening. Hopefully, maybe some drip screens or something like that that might be helpful in the long run for SDS. But this is certainly kind of a basic development idea. Okay, so I'll switch again really quickly, and I'm sorry I have not much more time to talk about the models. There were other things on models too. I want to talk a little bit about just what has been done and what's been progress with respect to patient populations, because that's a very fast-moving field. And in particular, I want to stress a couple of things that already several groups, and I know that there's ongoing work from the SDS registry, for example, looking at trying to find additional genes that might be the cause of SDS. Generally, there's a whole slew of papers that have come out looking at groups, and people have grouped their patients in different ways. I would say probably from the registry, it's a bit cleaner, I think. And what I mean by that, it's more SDS because these, by definition, are patients with an SDS diagnosis, whether or not you know the causal gene. So with the idea that if we could sequence all the genes, can we find new ones that also lead to SDS? This is not particularly easy to do, but it has happened. Or this is, in fact, one of the ways that DNA-JC21 was identified. It was by people looking at patients that looked like they had SDS, and then by sequencing them, it was able to identify new ones. And I've given one reference here of some of the older work that had been done because there's a lot of papers on this. One other feature, so hopefully this will help families that don't yet have do not yet have a defined cause at the genetic level. Another aspect of really lots of development have been in thinking about doing diagnosis, and I should tell you beyond this meeting, also thinking about it from the genetics perspective, there's now this idea that since sequencing, DNA sequencing is getting less expensive, and also there are other things to think about, just protocols and things like that, that would you consider to even do diagnosis by starting out with sequencing? Now, of course, you need to think of patient as an individual, and of course, you need to pay attention to clinical symptoms and presentation and such. But could we be a little bit more liberal in when and how quickly one goes to genetic testing? And this idea is really picking up in genetics. For example, there are clinics now that any kind of developmental delay or failure to thrive, they say, oh, well, let's go with sequencing and see what happens and what do we learn. And it's actually quite fascinating what we're learning. Part of this is because today, if you're going to do sequencing, it's expensive, but you can do 400 genes, you can do 600 genes, and they're almost the same price. There's not a big difference. So people have talked about cataloging, and there's all kinds of publications coming. I grabbed this one, came out last week from Korea. There was another one from India last week where what they're doing is grouping what they have different groups. So this particular one from Korea was inherited bone marrow failure syndrome. So basically they took all the patients, all the young patients they had that had some kind of hematology, some kind of failure to thrive, and they just sequenced. They did find a Shwachman patient. In fact, they diagnosed in this study a Shwachman patient. Patient was less than 4 months old, and they had not much evidence other than the patient wasn't growing very well, and there was some indication that the blood was abnormal, but they could make a diagnosis that quick. And it happened that was the youngest patient they included, and that patient had Shwachman syndrome, that they went on then to verify that, in fact, by the pancreas that there was a problem and so on. And so that's how quick that was. So I just want to emphasize how fast this world is changing, because it's pretty prominent now that we're going to start sequencing patients with perhaps less evidence that one should, just because one can do it. It becomes cost effective because of time that it saves and so on. And as I said, you still wouldn't want to leave that the patient needs attention in terms of other phenotypes and all of that, but it would get to diagnosis fairly quickly. And this is now becoming a favored protocol by some institutions. It does need the benefit of economy of scale. That is, it isn't practical to do 2 patients. It's more practical to do 200 at once and this kind of thing. But diagnostic labs can be set up like that. That's what they would do. So thought that was evident. And we heard, as I said, some of that kinds of screening from some of the presenters in terms of where things are moving in that general area of doing diagnosis and getting very quick diagnoses. I also want to mention there were a few other little interesting tidbits, though, that came up out of that. And one was that in some families, and I think we all have our anecdotal stories, I know I have a couple of experiences of appreciating that additional cases were found in families when there was 1 patient, but additional patients had been found that way. And I remember a couple of them happening in our original study too, as well, that either the patient was missed or the patient was younger or the patient appeared generally not as severely afflicted, at least not through the time point the data had been collected. But still, there were anecdotal stories of this from more than one group. There was also some interest expressed in the idea of doing newborn screening for SDS, and that would be the idea that you would screen for it from the beginning, with the idea that earliest intervention is the most helpful. And certainly that would be criteria to have newborn screened. I can tell you newborn screening and selecting of diseases for newborn screening is, I'm sure, complicated by politics and home and what the rules are in your given country, or I think in the US it's done by state and so on. So there are some social things there going on and political things probably that need some investment, but this is not that far beyond the horizon, with the idea now that in genetics we talk about, well, we should just sequence all babies and see if they're carrying severe diseases. So it's kind of merging a little bit, and we'll see where that takes us going forward. Okay, so this is for new genes and also improving diagnoses. I want to spend a little bit of time thinking about what you've all probably had a little taste of and heard a little bit about. And this is the idea. So just what is going on in the blood and what is the sensitivity of the marrow, the bone marrow in Shwachman syndrome. And I need to remind you that I'm not a hematologist, so I'm sorry if I get some of this wrong, but we had many talks, so I feel somewhat learned on this a little bit at least. So this idea of clonal hematopoiesis, we've actually known about it in a way in Shwachman for a long time. And I'm particularly thinking of those very old studies, very old, well, not that old, 30 years or so, where people were looking and you could see in the bone marrow and blood cells of Shwachman patients that there were these strange things going on with these chromosomes. There were chromosome losses. There were things chromosome 7 was doing. We see stretches of genes being deleted. But now with the newer methods of sequencing, we can be much, much more sensitive. In fact, we're finding many more things, not just chromosome losses. We're finding genes that have sequences that are changed. And what we're learning is that this can happen very, very early in time in a patient. In fact, in Shwachman, it happens very early. We know of clonal hematopoiesis being a major issue as you age. In fact, once you hit about 70, you have quite a few acquired clones in your blood, and this is just due to age and time. What is quite striking in Shwachman is that we're seeing this early. But let me tell you what I mean by a clone. So now we're looking at specifically at the hematopoietic stem cells. So these would be cells coming from the bone marrow, and we have a clutch of them here. What happens with time is that one of them changes, and that's what I've tried to depict with this gray to red here. Okay, so it's acquired a change in a gene. We'll come to the genes in a moment. Okay, so what happens with this cell? Well, with time in these stem cells, as they make more stem cells, they're going to carry this new change that happened, probably by chance, in a way, it happened by chance. But what you're gonna see is how this changes. This clutch is changing. We have more cells, but now we, as of course, by proportion, also have more cells with the red nucleus. Okay, we have more cells here with this change in the red gene. Okay, as time goes on and these cells continue to grow, we may see even more of these red cells. We could also anticipate that other cells might acquire another change. And here we have one with a little bit of a purple change. So these are different changes. When we're talking about a clone, we're talking about these cells with the red. That's one clone. This is a red clone. Okay? So all of your bone marrow cells, a portion of them are different, and they're different in the same way. So we're calling that a clone. Could also eventually from this have a purple clone. So this bone marrow would be carrying two clones, a red clone and a purple clone. And in each case now, red refers to one gene and purple is referring to another gene. What matters is what are these genes and how are they changed? And I'll come to that in a moment. But I want to emphasize a few things that if this occurs in a stem cell, and you know about progenitor cells in your blood lineages, that later the neutrophils and all the mature white blood cells come from the stem cells. So if some of the stem cells have this change, then some of the later cells are also going to have this change. And that changes things for the bone marrow and for what might be happening and what might be envisioned or what might be visual in the blood. That we have to think about that. Now, one of the things that you could think about is, well, when we talk about a clone, how do we measure it? So I want to introduce this idea of variant allele frequency. So we have a change in a gene and we know what it is. And you see, we're looking here at these 5 cells, and it's only 1 in 5 are the red cells. So you're going to measure a change in a gene. Well, that change is only going to be evident in one of the 5 cells. And since you mostly have 2 copies of every gene, that means it's 1 in 10 is what number you're going to get. So you're going to get a number like 10%. As this cell grows and becomes a greater proportion of the bone marrow cells, that variant allele frequency, the proportion that you're going to see that has this change is going to increase. It could also decrease if this red clone got taken over by other cells that are different and don't have this change. So it can change. But that's what we mean by variant allele frequency. But what we've learned that we really have to pay attention to are just what are the genes that are changed here. So we're going to talk about driver versus bystander genes. Well, driver genes are genes that is what is depicted here by this red, that you can see the red cell, the cell with the red nucleus, grew faster, presumably, than the ones with the gray. So that change in that gene is in fact a driver gene. So just looking at that alone doesn't mean that much. But what do we see when we start looking across patients and in several people, in the bone marrow of several people? We want to think about that. In fact, one thing that's very striking and seen in Shwachman patients, and you'll know some of this, that sometimes we have something called a 20q deletion. This was seen as a partial chromosome loss from many years ago. And now, not only do we see partial chromosome loss, we actually see that there can be variants within the IF6 gene so small that you have to actually do sequencing to be able to see it. But with the new sequencing tools, we can see it. Okay? And that can happen. This is, in fact, a driver gene because it turns out that we do know that once this 20q change has happened or a change in the IF6 has happened, that will tend to persist. So we'll get an EIF6 clone, that's what we'll call it, okay? It's a stem cell. It still is a stem cell that has the properties of stem cells, but it also now has a change in this particular gene. Of interest is this is being seen, and it's repeatedly being seen in multiple patients at different stages, but we can see it in the bone marrow. And the reason we pay attention to it as a driver gene is that, generally speaking, if a patient does have EIF6 clones, there's generally this idea that that gives a little bit of protection, that marrow will be less aggressive in terms of transformation to cancer. Not quite all figured out why that's true, but we know that. But go back and think about the slide I told you about when we mutated TIF6, which was the equivalent to EIF6, it sort of alleviated and let the yeast cells grow a little bit better. So we envisage that what's happening when you take away one of the copies of the EIF6 genes in humans in the bone marrow, are you giving that cell a slightly better chance to grow? Because now that even though SBDS is also missing, because SBDS is missing in all cells in a patient with SDS, and EIF6 is now missing in the bone marrow of the blood cells, have you given those stem cells a little tiny bit better chance to grow? And that's the suspicion. Okay, that's why we say this is its driver gene. So, and I'll come back to this in a moment. The other player that's very clearly a driver gene in Shwachman syndrome, and it's a striking observation that we're quite surprised with, is the occurrence of p53 clones. P53 is a gene we know really well, and it is a concerning gene. It's a concerning gene because we know that if you lose this gene, that there is a propensity to transform to cancer. And what's striking is that we see this very, very early in SDS. Of all the diseases that we know about, lots of changes that can happen in leukemia, and we know that you can have p53 clones. What is not common is to see it as early as we're seeing it in SDS. But even then, it's worrisome, but what exactly does it mean? Because you want to remember that we have 2 copies of every gene, and so you take 1 copy out of p53. Okay, we'd be more worried if you lost both copies. But with 1 loss, it's not clear what the outcome of that is. Okay, so we have, so I just told you about the growth. So you'll hear about the idea that p53, which is a known driver gene of transformation to cancer, we say that's a maladaptive fitness survival, because one of the concerns would be if p53's job is to slow growth, particularly when it senses there's something wrong in a cell, but you take that away, you're probably giving that cell a fitness survival, and it's not favored because this cell is not a happy cell, but he's got the property of he can grow a little bit faster. Well, growing a little faster is not always a favored event. EIF6 loss reflects adaptive fitness. That is, this fitness perhaps gives a little bit of survival, but it's giving survival because it's making the cell more tolerant of not having SBDS. Okay, p53 is kind of doing the same thing, but now he's just bypassing a safeguard, and that's maybe not the best thing to happen. But having said that, and being worried about the p53 that are early, we have to think very carefully how to interpret the change on 1 copy, and that's not so easy to do, to think about the change, because, for example, what has been seen, and there were presentations on this at the meeting, there's also been some publications on this, we actually realize that there are patients that have changes in their p53 genes that tend to persist for quite a long time, like years, and it's not clear. We do know to be a little bit more worried if both copies are compromised, so that's a little bit more concerning that we'd have to think about. But this brings up really a number of interesting questions that we have to think about. what exactly are the parents of the patterns of the arising clones and their persistence, how to interpret them, not absolutely straightforward. Are there other driver genes besides P53 in the cells of the bone marrow? And I'm not going to talk about them too much. There have been reports from both the Boston groups and with registry data, and also from England, from Cambridge and Warren groups talking about some of these events and what some of these genes could be. There are other genes. I want to bring back the definition of a bystander gene. We do know when we look at cancers and how they develop that there are genes that get changed that we're not always sure that contribute to the cancer, so they're called the bystander genes. You can usually tell the difference between a bystander gene and a driver gene because the bystander genes will tend to be more erratic, that is, they occur in some cases and not. But we know that P53 is a driver gene because we see this in a good portion of SDS patients. Sometimes quite early, but we don't, but as I said, we don't always know what it means. So certainly what is emerging, the idea here that's emerging, can we improve the surveillance of bone marrow? Because we know we have to pay attention to P53, but we still have to learn a little bit more about how to figure out what it's doing. We have to, and there was then some talks on thinking about this, on rethinking this. We know that the variability in Shwachman persists into adulthood. We know that there is increased risk with age, so that's something to think about, that we have to think about carefully in thinking about caring for patients. We're very excited to hear about an ongoing international study with multiple centers involvement where they're going to look at occurrence to leukemias and things like that. And I bring this up because although, there's clutches of 50 patients here and 100 patients here, we'd really like to have much bigger numbers, and that there is a study that's ongoing. We heard a little bit about that, so that was very exciting to hear about, and we look forward to hearing more outcomes on that. That's at a stage where it's being pretty well organized, and there's sort of plans for what they're going to score and things like that, and that was pretty neat. And but also want to go back and think a little bit about, so we have these really valuable clues here of seeing these driver genes that are happening in the bone marrow. A lot of questions left about what does it mean exactly, and how do we get the best information out of and use that information to help patients. But the one thing that we have, if we compare the development of cancer in Shwachman syndrome to the development of other kinds of leukemias, we actually see differences. And that's why it's so hard to figure out what's going on in SDS, because we know a lot about what happens in leukemia, but so usually what we do, if to look at leukemia and for progression to transformation, you look at the bone marrow, you see weird things in the numbers of cells, and you see weird things in how they look. They look weird, myelodysplastic syndrome. And then the prediction is that that will continue with transformation to leukemia. So that's generally for leukemia. That fits the patterns of how we think cancer develops in lots of tissues. You see this idea of progression, that there's an evolution. You see peculiar things, and then you see stages and steps that happen that you can score, and then you get a better idea of how that then leads to leukemia. The challenge, though, in SDS is that's proving to be really difficult because there was lots of discussion on how, while we're very good at looking at peculiarities in the blood cells in people with MDS or people going to leukemia in older ages and such, these things don't really apply in SDS. But we have a new player now. We have to think about the early involvement of P53. So we see early P53. We may or may not see myelodysplastic features. And the frustration is still true that the changes can be abrupt. And there's also evident that clones can persist for a long time without additional change. So although in some cases it transforms to leukemia, that doesn't happen in all the cases. And we certainly don't know why sometimes it does and sometimes it doesn't. It'd be good to know why it doesn't, because that would be a suggestion of, okay, how do we do this and how do we keep this not happening for all patients? But that's actually quite difficult at this stage. So we were looking then at thinking about, so what we've learned about what's going on with the bone marrow. So one of the concepts that was coming out, and you'll see this in the literature too, we see this for all cancers, where we think about, can we define high risk features that then you need to pay particular attention to, but we need to be able to define them. So certainly whatever's going on with P53 would be one such risk feature. This will help think about doing bone marrow transplant, because although bone marrow transplant is currently not your first favorite thing to do, it is, and if it's done early enough, seems to be helpful for some some patients. It's also true that bone marrow transplants are being more successful and with improvements in how they're being done, and we're sensing that. It's certainly things are looking better. We still need them to be better, but they're better than they were. So that's a positive. So currently, and this was, I thought, very nicely explained. I think this was a talk from Dr. Shimamura about, we think about when would you do a bone marrow transplant? Well, you could do it at sort of late stages of transformation, that is toward when the cancer is there, but this gives you somewhat limited things, versus a preemptive strategy where you do a very early bone marrow transplant, but we need to remember that bone marrow transplant is not without complications, and it's a high risk, has some risk in the procedure. But what about something in between? Could we come up and consider, are there a set of circumstances or situations when there are high risk features present? These could actually change with age, because we know the risk goes up with age. So you might change where these boundaries are. Could we come up with boundaries for what these high risk features are and use that to help to guide how to think about doing bone marrow transplant? And that was some of the discussion. That was certainly, people had somewhat different opinions on that, especially at deciding on the boundaries. But as we go forward, I do see that things can improve at that end. And we'll also have to remember that for bone marrow transplant for Shwachman, that some strategies and some treatment regimens we know are not favored and don't work, and these are been published and things, and so they're not recommended. So you want to always kind of remember that you want specialists to help you that are aware that you can't just use everything that applies to leukemia, because that does not apply to the development of cancer in SDS. So you want that to appreciate that and to know that. So on that, I'm just going to now conclude actually and give you some take-home messages, so a little bit of a summary. So I really jumped from all very or clinical stuff to very basic stuff to back to clinical stuff. Again, I think the most exciting thing about a meeting like this is, it's always good to meet people in person and to see families and meet colleagues and physicians. I was especially pleased to see many younger and new to field members at this meeting, physicians and researchers, and that was really exciting to meet people. And it's a very pleasant part of the meeting. We also had good chance to share issues and challenges of living with SDS, and we've reviewed those a little bit. Talked a little bit about causal genes and proteins. We're still looking for more, but we are learning more and more about the ones that do cause SDS, and that's actually very helpful in terms of, we're also learning a lot about ribosomes, which is, we know we need to think about that. We learned some, and I didn't have much time to talk about this, skeletal aspects, liver, gastrointestinal features. Mostly they were descriptions, generally speaking, but we need to think about these because there has been a request, certainly asking for research on these organs. And we also learned not just to think about younger people with SDS, we also have to think about age and what consequences there can be for different organs. There was presentation, which I didn't have much time to dwell on, was neurodevelopmental aspects and features, and there was some emphasis on ongoing needs, but in particular, remember that this is an area where maybe some remediation. So if you could capture vulnerabilities very early in behavior or in learning, and that would be a chance to help. So that was very interesting to think about that. Talked a little bit about it. I talked a little bit about SDS models, though I talked about organ on a chip mostly. But to remind you that we need to understand the failures of when things are disturbed, and remind you also that models really help you understand how to alleviate consequences, because these are important tools to test therapeutic strategies. So they are very important resources to have. And just finally, a few more things. There were ongoing studies to look for additional causal genes. There's new study activities of related genes, and I didn't talk about it so much, but that ZNF622 gene, which is a step just before SBDS, he plays a role in the DNA JC21, and that was interesting. I also kind of referred to this idea of the natural balance of proteins and protein levels in cells. I really look forward to seeing where that takes us, because certainly we know that growth is poor in SDS, and one tries to think of, and I like to think of it, that nature always figures how to balance everything. And whether you have if you have SDS, you have your balance. If you don't have SDS, you might have a different balance. That doesn't mean things don't work all the time. That shifted balance might just mean that you can't respond to a stressor as good as a person without SDS. There is a balance because nature always requires a balance. And we need to think about that because we don't always know all the players that could be involved. And I thought that was pretty interesting. More immediate, I think things I hope we're going to see are changes with regard to bone marrow surveillance. I hope I've hinted at my enthusiasm or my excitement at realizing more and more that maybe we can define things like what these high risk factors could be, and we could come up with boundaries for how we might change how to think about um, caring for what's happening in the bone marrow in Shwachman patients, and most important, because maybe we could be more proactive and not just react, because that would be a much favored kind of way. Also look forward to new strategies. I just barely alluded to the idea that can pharmaceuticals be developed? Can we think about gene and cell therapies? I didn't talk about this very much. There was some reference to that in different talks from the different presentations, but I didn't get a chance. And so that was what I was going to present. I'm actually happy to take questions, but I need to acknowledge a few people for sure. The hosting center at Cincinnati Children's and Myers and her team. I noticed the registries team was very involved in organizing the meeting as well, so acknowledge them as well. I want to thank all the speakers and the poster presenters, and I'm really sorry if I short-rifted somebody or misinterpreted, but I'm sharing with you what I learned at the meeting, and hopefully that was helpful. And I want also to acknowledge the various supporting groups for the meeting. And so I'll end here, but as I said, I'm really happy to take questions. So when I think the plan is, if you want to write the questions in the chat, I think that's how we're going to move forward. So I'm going to turn my slides off now.
And we do have a couple of people that typed in, so we'll start until people start to think of things. The first question is actually had to do with is there anyone who has had experience with tooth implant and specifically the difficulty in good bone formation after removal of native tooth, frequent tooth decay and poor enamel formation? I do know that, because there are bone issues with Schwachman, there are a lot of uh, tooth issues with Shwachman-Diamond. And I don't know anybody specifically, but I have heard of the poor enamel, not specifically with implants, but I guess if there's, bone issues in your jaw, you're going to experience difficulty with your teeth. Um, I don't know if
Yeah,
anybody else, Joe.
there wasn't
Go
so
ahead.
much at this meeting. We have had dentists before share some of the issues. Um, but yeah, and there are concerns for the enamel and things like that. I'm not sure. I think there's some publications. You could look up some of those for what has been found and how there was responsive. There was some proactive things you could do to try to help the sores that might be present in mouth and things. I don't know about implants, not a dentist, and but maybe
Yeah,
if
I'll
someone
see
can
if I
talk
could
more
find some of
more
the
informed,
articles.
yeah,
Yeah.
yeah,
Yeah.
that would be.
Um, so we have for someone who was clinically diagnosed 20 years ago, presenting as heterozygous for SBDS, is there significant genetic advancements that could more accurately diagnose her issues? Do patients now have access to more detailed genetic diagnosis?
Um, so sorry, there was, okay, let me see if I can see the question exactly. Can I,
Yeah, if you open your chat, you might be able to.
where is my chat under here?
It's usually well, mine's at the bottom, like with the,
Oh, there it is. Sure. Yep, it's coming. Okay. For someone who's clinically diagnosed 20, presenting as heterozygous with SBDS. Okay, interesting question. Might want, so at this stage, there is a clinical diagnosis. There is one very heterozygous. Um, I know what I would do. I would retest SBDS. I'll tell you why. Because it's one of the hardest genes, and diagnostic people call me all the time and complain about why you find the gene that's the hardest to study. And that's because there's a pseudogene that looks just like SBDS, and it complicates sometimes the interpretation. So you need a diagnostic lab that's pretty hot on that. In fact, we did hear at the meeting of people who went back and got a genetic diagnosis, and they did get an SBDS variants were found in their genetic material that initially were not, and that had to do with the analysis that was used to interpret the sequence. And part of that was the time, and the newer tools are now better. So that might not be an idea. That may be an idea to think about. Depending on where you are, there's different ways. You need to get a genetics, somebody who can order a genetic test if you wanted it redone, or you could participate in a research study. Even contact the SDS registry and they could help move things forward for you. That's what might be something I might suggest if that works. To see what they recommend, because it also depends where you live and all kinds of things, how to go about and get that consult.
Next, yeah, does anyone have reactive thrombocytis? I don't know of anybody, but I'm not
No,
familiar with that.
not familiar either.
Yeah, we will, check some of these. If we don't have answers, I'll check further and we'll see what we can do. What new meds are being seriously considered is a question.
Um, So I'm not perhaps the best person to ask because I'm not a physician, I'm a scientist. I did glean from the meeting, though, that it kind of depends what the meds are for, but certainly in the bone marrow transplant field, for example, there's certainly improvements coming and different procedures that are being worked out for regimens to prepare for bone marrow transplant. There are treatments, and these are not new, but there are different formulas, I think, that are being prepared for treatment of neutropenia. I don't know if they apply to SDS, though. I've just heard about that. That they were coming, there's some coming along because, they make treatments, they make short acting and long acting, and there's all kinds of things like that going on. That's a question that I suggest that maybe to ask um, probably ask hematologist, your hematologist, or might throw that one at the SDS registry too, because I'm sure they
Registry,
get
yeah.
that question. I'm sure they get that question once in a while.
Yeah.
Um, oh, and Mary is saying send info. Mary will send some info.
Yeah, we'll respond with some of these with info.
Yeah.
Yeah. Is there more updated research on GCSF? That,
That I don't know. I know they're changing the formulas to make it more and less potent. I don't know exactly. I don't know of any that wouldn't have been the topic here at this
yeah,
meeting,
it
but
it
it
wasn't,
would
yeah.
be from the neutropenia literature, and there would have been experts who would have been able to answer, although that's not me. Some of the neutropenia experts would have been up on where that's going. What is the end? So what is the anticipated timeline for gene therapy for SDS? So I didn't have time to talk very much. There was some presentation on gene therapy for SDS. In some ways, I think SDS is a good candidate for gene therapy, but that's not a minor undertaking. It does take years to develop. Um, we need evidence and we would need more work to know that that's practical. We probably need to try it in an animal model, for example. I'm not sure if there's any company working on that right now. There probably are a couple, but I wouldn't necessarily know which one or because they don't always disclose what they're working on. The sickle cell treatment, yeah, is now out there. It's not being used for everybody, but there are now some patients that are getting sickle cell gene therapy. it isn't, not everybody's a good candidate either. So these are things that need to be thinking about. I don't know what the timeline might be. There are other approaches to thinking about therapies too, though, that might be considered. Um, let's see here. I'm not sure if I can find anything on gene therapy or who would know. Mary, I'll try to
Yeah.
find something and send it to you, and then maybe if you could
Okay.
contact and see if there's action on there. I'm not aware. Like people have talked about it, but people were still talking about how to optimize to do it and things like that. I would say it's not
Yeah, it was
trial or
it
something.
was it was it was spoken about, but not really in
Right.
depth.
It was well, and it was spoken about from the context of how to deal with this gene and how
Yeah,
to make it work, not so much to do it directly, because
right.
that's always a
Yeah.
later step. That's always a later
Yeah.
step. Um, Do the
So
findings regarding being early indicators of problems developing mean blood analysis can be performed in lieu of traditional annual bone marrow? Okay, so that's a really good question. And that is a question that is being pursued at the moment. They are trying to test whether or not, when you look at clones, can you use blood in bone marrow? And there are some people who argue you can already. I know that in other diseases they say you can, but you formally have to look, and that study is currently underway. And one idea might be not so much that you would do away with the bone marrow, but maybe you would do it less and intermittently you would do bone and blood analysis. I kind of see that as a favorite approach, but that needs to be worked out. I talked a little bit about that variant allele frequencies, and I can tell you that when you look at bone marrow versus blood, they don't match exactly. Although I do know of cancers where they are now able to do blood and not tumor tissue, for example, it's getting that sensitive. So I think that's a route that's being followed because obviously people would favor that. The other advantage of being able to look at blood, it might be more, or it might be more feasible to do more screen, like more often as screening because it's blood, it's not bone marrow. It's not as difficult to get access to and things like that. So certainly that's being worked on. Um, oh, we have a CF question. Oh, as a carrier, heterozygous in addition. Well, so I'd have to look up this particular variant. Heterozygous in addition to SDS. Need to remind you that the carrier frequency of CF is about 25 times higher than it is for SDS. So if you have a big enough denominator, there will be people with SDS that are a CF carrier. That would be the case. I'll have to look this up. Maybe if you could give Mary your contact and I can look at it a little more closely and send an answer. Could it have implications? We don't think so. I would not think so. Okay, just by thinking about it. And that's because 5% of people walking around in this audience, there are probably people in this audience who are a CF carrier. You can be both a CF carrier and an SDS carrier. There will be some small portion of the population where that's true. Um, I don't think it has implications. And I need to check that variant. I need to be sure it's causal, and I'm not sure. But I can look it up. It's a valine to methionine. That's usually not a major chemical change. So that's why I'm suggesting I'm not sure it's going to cause anything. Um, Okay, is rickets and or Ehlers-Danlos syndrome commonly seen with SDS? Um, ooh, that's
I
too
know that I know that vitamin D deficiency
is, yes.
um
And that's not
definitely
rickets
is.
anyway,
Yes.
because rickets is vitamin C deficiency. But vitamin D deficiency is a concern because it's a fat-soluble vitamin. And if you are deficient in pancreatic enzymes and not getting enough nutrition and not digesting fat, you can have a vitamin D deficiency. Having said that, isn't the third of the population these days vitamin D deficient?
Right. Yeah. We're all supposed to be taking extra.
Yeah. But it is, you got to remember, you got to take it with fat, okay? Because it needs
Hmm.
fat to absorb. It isn't soluble in
Yes.
water.
Hmm.
So that's not a bad word, okay?
Yes. And everybody has to remember when you take your vitamins with SDS, you need to take it with your enzymes, or you're not doing any good. It's just going right out.
And that actually is the same treatment as used in cystic fibrosis.
Yes.
You need the
Yes.
ADAC vitamins and you need pancreatic enzymes as well.
Hmm.
And in fact, that's how that got learned for SDS. It came largely
Hmm.
from
Yeah.
Ehlers-Danlos syndrome is a different gene, different disease. Should not, I would not expect it commonly to be seen with SDS because it's It's more common than SDS, so yeah, there's some remote chance, but it's a different genetic disease.
Okay, that seems to be the end of our questions. If anybody had anything else, I want to thank Dr.
Oh,
Rommens.
there is one more question, and that as we
Is there
talk
one? Oh, yeah,
about
just popped
renal
up.
success, I think, well, I'm not a hematologist, like I said, but certainly from the meeting, one gets the idea that what you want is a place where there are experts in the field, and just to know that for a rare disease like SCS, and I know it's happened here, when we have patients, there's doctors consulting with each other to find out what to do. And so that would always be recommended that there's specialists that are familiar with SDS. They know about SDS because you want for bone marrow transplants. I told you there was some drugs that we know are not indicate that are counterindicated. They might work on older people with leukemia for different reasons, but not due to SDS. So you need that expertise. But obviously, what you can do, and the doctors do share. I know here to make a decision to do bone marrow transplant, I'm sure that Dr. Myers and Dr. Simramoro might get a phone call without any hesitation. I know that happens. That's just done to try to optimize and to do the right thing. And I think that happens
Yeah,
everywhere.
what we've seen is, yeah, if as long as you're working with the specialist, or at least calling your doctor is in touch with that, our specialist, um, the hospitals are, being successful it's when they're not specialists that we see the decline.
Right. There's
Incidence.
another question related to autism and SDS. Is the incidence known? No, not exactly, but it has been reported. And I know that because Dr. Elizabeth Kerr works here at our hospital, and I talked to her sometime about SDS, and she's done some studies on patients with SDS, and some came with a diagnosis of autism. And I don't know the number because that was a study of 30 people, and I don't think one could actually determine the correct number out of that small number of people. And in fact, she spoke at the meeting and talked about how we need to think about looking at more patients to see, and also looking at exactly the nature of the autism, and where are those vulnerabilities, where are the challenges, because autism is a pretty widespread definition that they change every now and then, and you'd want to look at that. It does. I somewhere think it was between 10 and 20%, but that was off 30 patients, so that's not a fair number. It certainly does co-occur.
Mm-hmm. Yeah, I think. All right. Anybody else? Oh, of course. We got two more now.
Okay, should a patient with go for bone marrow now that early renal coincided indeed to be a timely intervention? If I'm interpreting that question, I mean, a bone marrow transplant is a big decision. It's a big decision for you. It's a big decision for the family. It's a big decision for doctors to make, and that would all be done, circumstances, what are the high risk factors that are present, that needs to be decided as the team, and the team of your family and the team of your doctors. Yes, generally there's going to be a little more indication not to wait too late, but Dr. Shimamura had quite an explanation on this, on we need to think about and what are the boundaries of when is going to be the best time. Probably not absolutely clear, but certainly would look at age, would look at circumstance, would look at family, would look at other issues, and such. Oh, and I see that Mary has responded to consider Cincinnati Children's, and they have a fair amount of experience indeed.
Yeah, and Virginia, it's far. It's a ride, but not too far to go there. So yeah.
So question P53, an indication for early bone marrow transplant. My understanding is no. No, at least if the variant allele frequency is quite low, and that's because they have seen patients that show clones and they can have them for years, and not change. I think I have the sense that One would be more reactive to seeing changes in things. If something is static, they don't get too worried. That's my feel for how a hematologist react. But you need a specialist. Maybe I'm not that person. There's a question, do they diagnose clinically or with genetic testing? Yes, both. Yeah, I mean, always ultimately it's clinical diagnosis, no matter what. Genetic testing confirms. Um, okay. Okay, so if there's any more questions, I can have them sent to me, and at a later time or something. And
Yeah, we recorded this, we'll put it out there on YouTube. We'll let everybody know who was at the meeting in case you want to watch it again. If you think of any questions afterwards, please just send us an email and we'll get it out there. I know there were some that we said we would um, get back to some information on, so we'll take care of that. And we'll be doing hopefully some more of these, trying to get some more doctors on here to talk to you guys. So we're going to try to keep this up. Mary, you got anything else you want to say? She's got one of the little ones.
No, but I appreciate everything. Dr. Rommens, of course, you did a wonderful job again. Thank you. You too, Deanna.
No problem. All right. Thank you all. Have a good night, and we'll be talking soon. Promise.
okay, bye
Bye.
everybody.
Bye.
Enjoy the rest of your summer.
Yeah.
Thank
Yeah,
you very
not too much
much.
sun.
Yeah, keep out of the sun. Your P53 gets activated. Bye-bye.
Bye.